Curious. Hello, and welcome to another episode of Science Simplified, where we explain complex scientific concepts in an easy and fun way. Today, we're going to talk about recombination theory, which is a branch of genetics that deals with how genes are shuffled and exchanged during reproduction. Recombination theory is based on the idea that genes are not inherited as fixed units, but rather as segments of DNA that can be rearranged and combined in different ways. This process creates new combinations of genes that can result in variations in traits and characteristics among offspring. But how does recombination happen? Well, there are two main mechanisms that are responsible for recombination: crossing over and gene conversion. Crossing over is the exchange of DNA segments between two homologous chromosomes during meiosis, which is the type of cell division that produces gametes or sex cells. Homologous chromosomes are pairs of chromosomes that have the same size, shape, and genes, but may have different versions or alleles of those genes. For example, one chromosome may have a gene for blue eyes, while the other may have a gene for brown eyes. During meiosis, homologous chromosomes pair up and align along their length. Then, they break at one or more points and swap corresponding segments of DNA. This results in new combinations of alleles on each chromosome. For example, a chromosome that had a gene for blue eyes and a gene for curly hair may end up with a gene for brown eyes and a gene for curly hair after crossing over. Crossing over is a random process that can occur at different locations and frequencies along the chromosomes. The closer two genes are on a chromosome, the less likely they are to be separated by crossing over. This means that some genes tend to be inherited together more often than others. This phenomenon is called linkage, and it can be used to map the relative positions of genes on chromosomes. Gene conversion is another mechanism of recombination that involves the copying of DNA from one chromosome to another during meiosis or mitosis, which is the type of cell division that produces somatic or body cells. Gene conversion can occur when there is a mismatch or error in the DNA sequence between two homologous chromosomes or sister chromatids, which are identical copies of a chromosome produced during replication. When this happens, one strand of DNA may act as a template to repair the mismatch on the other strand. However, instead of restoring the original sequence, the repair may copy the sequence from the template strand. This results in a change in the allele on one chromosome or chromatid. For example, a gene for blue eyes may be converted into a gene for brown eyes by gene conversion. Gene conversion can also occur between non-homologous chromosomes or DNA molecules, such as mitochondrial DNA or viral DNA. This can result in the transfer of genetic material between different species or organisms. For example, gene conversion may be responsible for some cases of horizontal gene transfer, which is the movement of genes across different evolutionary lineages. Recombination theory is important for understanding how genetic diversity and evolution occur. By creating new combinations of genes, recombination increases the variation among individuals within a population. This variation provides the raw material for natural selection, which is the process by which organisms with favorable traits survive and reproduce more than those with less favorable traits. Recombination also allows for the generation of novel traits and functions that may enhance adaptation to changing environments. Thank you for watching this episode of Science Simplified. If you liked it, please give it a thumbs up and subscribe to our channel. And if you have any questions or comments, feel free to leave them below. Until